Hi, welcome to my talk on the machine learning-based methods DeepViral prediction of virus host protein-protein interactions. This work is jointly supervised by Professor Jasper Tegner and Professor Robert Horndorf. It was recently published in Bioinformatics earlier this year. The code and data are both available on GitHub and Zenodo. Viruses, unlike bacteria, cannot replicate themselves. Instead, they hijack the whole cellular machinery for their replication through a complex network of interactions between proteins, RNA, and DNA. Here's an example for the SARS-CoV-2 virus that gave us the COVID-19 pandemic. Scientists look at the protein expression levels of the interacting human proteins across different tissues, and they find out the highest expression is in the lung, but it's also highly expressed across many other important tissues. The prediction of protein-protein interactions, or PPI, have been attempted through many different machine learning models, such as SVM, random forest, and neural networks. However, these are only with protein sequences as features. And in our study, we're proposing to use phenotypes, the infectious disease symptoms, as an additional feature for their prediction. So here on the right here, we have some example phenotypes for the influenza C virus. Their general symptoms, fever and cough, but also more specific phenotypes that can help us distinguish between the differences of different viruses. And to use phenotypes in machine learning models, we need to convert them into numerical representations or vectors through some methods called embedding methods. And here we use a recently developed machine learning embedding methods based on random walks and word-to-vec. At the end, we convert for each human protein and each virus species into a feature embedding of dimension 100. And this is our model architecture. Our model takes as input a pair of human and virus proteins. And for each protein, it uses its phenotype embeddings and the protein sequences as features. And at the end, the model outputs a 0 to 1 score, indicating the likelihood of interaction. And this is the evaluation results of our model DeepViral against existing methods across 14 virus families. And we call this a leave one family out evaluation. And the DeepViral model, based only on sequences alone, performs as well as the existing methods. But after adding in the phenotype embeddings, which we call the joint model, DeepViral performs the best across all the metrics. We also showed that this is also possible for individual species, and we call this a species level optimization, or leave one species out optimization. And we showed this for 4 important virus species, including SARS-CoV-2. So as a conclusion, phenotypes and features do improve prediction performance. We also discussed how to evaluate machine learning methods for novel virus protein interaction prediction. And we also discuss, in order to improve for a specific virus, the prediction model needs to be adjusted. For example, SARS-CoV-2 can potentially benefit from adding in the training data of its taxonomic relatives, SARS and MERS. This is at the end of my presentation. I want to thank my advisor Jasper Tegner and Professor Robert Horndorf for their supervision and the support from my groups. And I also want to thank the supercomputing resources from KAUST, which made this work possible.